Hello, students. This is Dr. Faizan Mirza. I'm discussing with you the questions regarding Hardy-Weinberg principle. The calculation of Hardy-Weinberg principle is what I'll be covering here. I think I'll be taking two questions here. So let's start. So the question is that the Hardy-Weinberg principle may be used in calculating allele frequency and genotype frequencies for a gene within a population. The Hardy-Weinberg principle use these two equations. We have the two equations. Although we don't need to use the first equation, we can just use one equation and we can get all the answers. So within a population of butterflies, the allele brown wing color capital B is dominant to the allele for white wing color, which is small b. So we know that all browns are capital B, capital B, capital B, small b, either one of these, and whites will be small b, small b. 40% of butterfly in the population are white. So it's always how do we count the phenotypes? We always go for recessive one. We count the recessive in the population. So we know that how many recessive are there in the population. With that, we can calculate how many of these can be homozygous dominant, how many of population can be heterozygous dominant. But we need to know the quantity or the number of the homozygous recessive in a population for any trait whenever we are doing Hardy-Weinberg principle. So this is already given to us that recessives are given, and they're asking you to calculate the percentage of butterflies in population that are heterozygous for the gene controlling wing color. Now we need to just recall what is p square. P square is the dominant alleles present in a population, whereas a single p represents a single dominant allele in the population. Similarly, q square represents the recessive allele in a population, whereas a single q represents a single recessive allele in population. So square and non-square actually represent the alleles in population, and without a square, it represents the allele in population, the single allele in a population. Similarly, when we are using 2PQ, 2PQ represents heterozygous condition. So moving on from here, we know that homozygous recessive given to us is 40%. So this information is given to us as Q square. So 40% is Q square. So we have 40% Q square. So we'll just calculate Q square as 40 upon 100. We'll convert this into a frequency. So from percentage, we calculate the frequency. So we have frequency. How frequent is it? This is 0.4 frequent. Its frequency is 0.4. So what about the single recessive allele? Q square is 0.4. So for single recessive allele, we need to just take an under root of the value of Q square. The value of Q square is 0.40. So we'll just place it here. We'll take an under root of this, a square root of this, and we get 0.63. The value of Q is 0.63. So we know Q square was a small b together. This was 0.4, and single recessive allele Q, which is single b, is actually 0.63. Now, when we have the single recessive allele, what do we need to do? We'll calculate the single dominant allele. The single dominant allele will be calculated using the formula P plus Q is equals to 1. We'll just rearrange the formula for P. We'll take it 1 and we'll just subtract Q from 1. We just take Q on the other side. So 0.63 when taken here and subtracted from 1 gives us 0.37. So we have the frequency of the recessive allele and the dominant allele as 0.63 and 0.37. What we need to go, we need to calculate the heterozygous. Heterozygous is always 2p. So how do we do? We'll just apply the formula of 2 multiplied by p multiplied by the value of q. So we know the value of p and the value of q is 0.37 and 0.63 as calculated by us. So we get the answer of 0.466, which actually is the frequency. Now we just convert it to a percentage. So 0.466 means 46.6%, and we can round it off to 47% value. So this is how this question can be done. For situations in which Hardy-Weinberg principle does not apply, does not apply. This is just a fact that we need to just learn. It doesn't apply when there is a significant selection pressure against one of the allele. It cannot be applied when there is migration occurring in and out of population. It cannot be applied if mutation occurred and a new allele formed. It doesn't apply when the mating is not non-random. So you can state any four that you have already known. Let's take another example here. This question goes with selection evolution, and goes with the genetic the genetic technology as well. So the rainbow trout, the name is given in binomial nomenclature. This is the genus, this is the species. Is a fish that is bred in commercial farms. Rainbow trout that have blue silver color are sold at a higher price than rainbow trout that have brown color. So there are two types of rainbow trout: the blue silver and the brown color. There are two phenotypes here, and this again is an example of polymorphism. You can just recall what polymorphism is. It is just the occurrence of more than one phenotype of an organism in a particular population at the same time. So the number of fish with each of different color was recorded in a breeding population, one fish farm. So if it's a fish farm, they must be going for selective breeding. The total population of the fish is 2936. Number of blue and silver fish is 1437. Is it dominant? Is it recessive? We'll go for it in the information which is given to us later on. The number of blue silver is 1437. The number of brown fish is 1499. So this information is given to us. Now let's read further. The color rainbow trout is controlled by a single autosomal gene. What do you mean by autosomal gene? Autosomal gene means that the gene is located on the autosome and not the sex chromosome, with two alleles, one dominant and one recessive. So this is an example of a single gene, single allele gene. So this gene is controlling this is single gene with two alleles. So single gene, not single allele, single gene with two alleles, one dominant and one recessive. The blue silver color in a fish is homozygous for recessive. So we know with this sentence that blue silver is a recessive trait. So if we take blue silver as small b, so that means the recessive in population 1437, and the remaining 1499 are brown fish, which could either be capital B, or capital B, small b, either. So we just annotate this for our own help. The formulas are mentioned to us. Again, we'll use this formula. This formula, I think we can just avoid using this formula. Without this question can be done very well. Using this formula to calculate expected number of brown fish in population are homozygous. So they are expecting you to calculate capital B, capital B. Now we know that homozygous in our population is always p square. If they were asking for a single allele, we would have calculated p. But they are not asking for p, they are asking for p square. Although we have to calculate p and then go to p square, let's go with the calculation. So what is the frequency of population? Frequency in the population for the recessive trait. We know that the total population is 2936, out of which 1437 have recessive alleles. So we'll take 1437 and divide by 2936. We get the frequency, and the frequency is of q square, because this always, which is the population which is recessive, is always q square. This q square is given to you. So we have q square, we take out the value in frequency and we'll just convert it to a percentage. So this is the q square value. Now this q square can be converted into a single q by taking the square root of this value. We get the answer of 69.9% or 0.699 respectively, either for the percentage of frequency. Now, how the single dominant allele can be calculated? Single dominant allele, we use a formula p plus q is equals to 1. We'll just rearrange the formula in such a way that we'll calculate it for p. So we have the formula here, p is equals to 1 minus q. So 1 is, we'll subtract 0.699 from this. 0.699 comes here, we calculate from q. So we just subtract it, we get 0.301. Now with the 0.301, what do? We have the p here. Now we'll just take the square of this. We'll just square the value of p. We'll get 0.0906. This turns out to be 0.091, which actually is 91%. So we know 91% of the population is homozygous dominant. Now we need to calculate, the question is actually about this population. So the total population, the frequency is 0.091, and the total population of fish is 2936. We have to calculate how many of these are capital B, capital B. So capital B frequency is 0.091. So when we have 0.091 here, what we will do, we'll just go and we'll multiply this with the total value of 2936. We get the answer 267. So 267 are homozygous brown fish. So just the scientists, when this condition met some scientists suggested that Hardy-Weinberg principle did not apply in this situation because the rainbow trout being kept in a commercial fish farm, state two condition that must be met by the commercial fish farm for the population for Hardy-Weinberg principle to apply. There is no migration in and out of population, mating is occurring random. So if these conditions are met, yes, it can be applied. Describe how selective breeding can be used. Selective breeding can be used to increase the proportion of rainbow trout with silver brown, silver blue color in the fish farm. We know silver blue is a small b recessive trait. So what will we do? Selection pressure is exerted by humans, since this is a selective breeding process, to produce offerings of desirable traits. Dark brown rainbow trout is bred with shiny blue variant. Over several generations, shiny blue offsprings are selected and bred together, because when you have small b, you cross them with small b, offspring 100% will be small b. So the allele frequency in such selective breeding would favor shiny blue, and its allele frequency would start to increase since the recessive allele shiny blue will be passed on more as compared to the other. So you get 4 marks. Now they take the question to genetic modification. The Atlantic salmon, again, the name is given as genus and species, can be genetically modified. Genetic modified salmon are bred commercial fish farm for food production in 2017. GM Atlantic salmon, bred in Canada, became the first GM animal to enter the human food chain. I have discussed this in one of my videos in the chapter of genetic technology. Explain how this genetic modification of Atlantic salmon can be used to increase food production. So allele growth hormone is inserted for faster growth in salmon, and the promoter is taken from the other fish. They are tied together. The growth would occur around the year, because we know that salmon doesn't grow the whole year. It grows only in certain season of spring and summer. But if you insert this in gene of growth hormone, it keeps growing around the year. It would grow faster and a larger size. It would reproduce more, so its increasing its quantity would serve increased food production. So I hope this was helpful. Do mention in the comment section if you found this helpful. And that's it from my side. Thank you so much.